Good day, this is Professor Will, CBMD, PhD. Today's Sunday, December 9th, 2018. It is 18:28 and 44 seconds Eastern Standard Time. Science News. Revolutionary technology pinpoints biopsies to detect prostate cancer. Date, December 5th, 2018. Source, University College London. Summary, medical software that overlays tumor information from MRI scans onto ultrasound images can help guide surgeons conducting biopsies and improve prostate cancer detection. A team of engineers and medical researchers found that the technology enables surgeons to pick up clinically relevant cancers that were missed when using current visual detection methods. The best approach would be to use both technologies in tandem, according to the findings published today in European Urology. The software is deployed via a system called SmartTarget. The advent of MRI-targeted biopsies, where MRI scans are used to inform surgeons where a tumor lies before they conduct a biopsy tissue sample, has improved detection rates to close to 90% from 50% in the last 5 years. Now the SmartTarget system has further enhanced the technique by allowing a 3D model of the prostate and cancer to be created for each patient from their MRI scans using advanced image processing and machine learning algorithms. During a biopsy, this model is fused with ultrasound images to highlight the area of concern, which otherwise does not appear in the ultrasound images, helping to guide the surgeon while conducting the procedure. Until last year, when MRI targeting was introduced, the established way to test for prostate cancer involved taking a biopsy from the prostate without knowing where in the prostate a tumor was likely to be, resulting in close to half the life-threatening cancers being missed. Prostate cancer detection has been improving at a very fast rate in recent years, and this technology pushes the science forward even further, enabling clinicians to pick up prostate cancer quickly so that patients can access the right treatment early enough. For the present study, 129 people with suspected prostate cancer underwent 2 biopsies, one using the SmartTarget system and one where surgeons could only visually review the MRI scans. The 2 strategies combined detected 93 clinically significant prostate cancers, with each of them picking up 80 of these cancers. Each missed 13 that the other method picked up. The researchers say that the surgeons visually review the MRI scans should be used in tandem, with SmartTarget as using the technique enables surgeons to learn to make subtle adjustments such as adapting to the movement of the patient and the prostate as the needle is inserted. We developed the SmartTarget system to equip surgeons with vital information about the size, shape, and location of prostate tumors during a biopsy that is otherwise invisible on ultrasound images. The software provides them with a clear target, as MRI-targeted biopsies require a very high degree of expertise and experience. We hope that the imaginary displayed by SmartTarget will help to bring high-accuracy prostate cancer diagnosis to a much wider range of patients and hospitals. The researchers say the new methods could reduce the number of biopsies needed and reduce the unnecessary surgeries caused by overdiagnosis of less harmful cancers. The SmartTarget software has been commercialized by SmartTarget Ltd., a company spun out by UCL's commercialization company UCL Business plc, UCLB, and the system is already in use by several hospitals in the UK and USA. The interdisciplinary study brought together engineers, urologists, and radiologists supported by UCL Translational Research Office and Project Management in navigating the translational regulatory pathway involved in taking the project from the lab bench to the operating room. There has been much discussion and speculation in the media recently on the degree to which computers and artificial intelligence will be integrated into clinical care. Studies such as this one are extremely important as they provide valuable evidence on the performance of the new technology in the clinical setting. With this study, we now have hard data showing that SmartTarget is as good as a group of experts in targeting tumors in the prostate, and have a glimpse of how clinicians and computers will be working together in the future for the good of the patient. The date is Professor Will, CBMD, PhD. Today's Sunday, December 9, 2018. It is 18:48 and 19 seconds Eastern Standard Time. Science News. Scientists design a way to track steps of cells development. New tool has potential to boost regenerative medicine cancer research. Date, December 5, 2018. Source, Washington University School of Medicine. Summary. Scientists have developed a new tool described as flight data recorder for developing cells, illuminating the path cells take as they progress from one type to another. This cellular tracking device could one day help scientists guide cells along the right path to regenerative certain tissues or organs, or help researchers understand the wrong turns some cells might take on their way to becoming cancerous. Scientists hope to one day be able to take skin cells from a patient who needs a liver transplant, for example, and guide the skin cells along a known path that will result in a new liver. Without this cellular tracking device, researchers are able to study the original cells and the final cells in great detail, but the pathways cells take to reach their destinations have been largely unknown. There is a lot of interest in the potential of regenerative medicine, growing tissues and organs in labs, to test new drugs, for example, or for transplants one day. But we need to understand how the reprogramming process works. We want to know if the process for converting skin cells to heart cells is the same as for liver cells or brain cells. What are the special conditions necessary to turn one cell type into any other cell type? We designed this tool to help answer these questions. According to the researchers, the tool could reveal cellular reprogramming, routes that might involve reverting skin cells back to different types of stem cells that could then mature into a new liver or other vital organ. Among many potential uses, the tool also could be applied in cancer research, recording the wrong turns normal cells might take to developing the tumors. Using their flight data recorder, the researchers performed experiments that uncovered some surprising details about the specific routes taken by cells that successfully completed their flight paths. The new information may help researchers identify the best pre-flight conditions to expose the cells to in order to increase the likelihood that their journeys will be successful. The scientists want to follow the right flight plan, so to speak, so all the cells arrive at the correct destination. Right now, cell reprogramming is really inefficient. Research says, when you take one cell population such as skin cells and turn it into a different cell population, say intestinal cells, only about 1% of the cells successfully reprogram. And because it is such a rare event, scientists have thought it is likely to be a random process. There is some correct set of steps that a few cells randomly hit upon. We found the exact opposite. Our technology lets us see that if a cell starts down the right path to reprogramming very early in the process, all of its related sibling cells and their descendants are all on the same page, doing the same thing. The technique harnesses the natural properties of the virus that inserts tiny DNA barcodes called cell tags into each cell. As the cells divide, their unique barcodes are passed down to all their descendant cells. At several set time points during the 28-day cellular reprogramming window, new barcodes are added and a sample of the cells are analyzed to see what they are doing at that waypoint. The cell tagging technique keeps track of which cells share common ancestors and how far back that common ancestor is found in the lineage, like a family tree. Indeed, beyond single flight tracking, the tools let researchers and the teams build complex family trees of cells where successfully reprogrammed cells can be traced all the way back to their early ancestors. Researchers say their study suggests that the state of the cell is in at the time it receives the instruction to reprogram has already set the stage for whether it will be successful or not. This contradicts assumptions that a cell goes in many different directions when it is first instructed to reprogram. If we can unlock that cocktail of initial conditions that prime cells to be successful in their reprogramming, we can transform cells into the type we want with much higher efficiency. We'd like to reach 100% efficiency. That would be really exciting for the field of regenerative medicine. Researchers already may have identified one ingredient in the cocktail. They found that if a certain gene called METTL7A1 was turned on in cells, they were 3 times as likely to successfully reprogram compared with cells in which this gene is inactive. According to researchers, another interesting finding was that the cells that were not successful in their reprogramming didn't just end up all over the map. They appeared to converge at the same dead end, tending to revert back to look like the original cell type. We see a single dead end right now, but as the technology improves, we think there is a possibility we will see multiple outcomes. The research studied mouse skin cells, exposing them to a molecular cocktail that triggers the DNA of the cells to open up, turn on new genes, and reprogram skin cells into another cell type called induced endoderm progenitors. Such cells give rise to both liver cells and to cells that make up the small intestine. In one example of potential application, researchers want to grow mini guts to help the study of premature infants with a condition called short bowel syndrome. Babies born too early are at high risk of disease that results in the death of tissue in the intestine. To stop the damage, some babies require surgery to remove that portion of the bowel, resulting in a shorter bowel. Since part of the intestine has been removed, these infants lose their ability to absorb nutrients. Researchers say if we eventually can grow a replacement intestine from the human version of these cells, That would be an amazing advance. This is Professor Will's CBMD PhD. Today, Sunday, December 9, 2018. It is 19:16 and 23 seconds Eastern Standard Time. Science News. Without DNA2, genes can jump into DNA breaks. Date December 5, 2018. Source Baylor College of Medicine. Summary. When DNA is absent, small DNA fragments jump from all over the genome into chromosome breaks. This novel mechanism may explain similar events commonly seen in cancer or during antibody diversification. One of the interests of my lab is to understand basic mechanisms of DNA repair. In this project, we worked with yeast and discovered for the first time a mutant that shows frequent insertions of DNA fragments into DNA breaks. This mutant lacks DNA2, an enzyme conserved among all organisms. Mobile genetic elements called transposons are known to jump from one chromosome to another. Research colleagues found that any piece of any chromosome can jump into a DNA break. During DNA synthesis, occasionally long-term strands of DNA can form and are normally eliminated by DNA2. In mutants that lack DNA2, these oversynthesized fragments can get caught in DNA breaks, causing genomic instability, which mostly has negative effects on the cells. However, these insertions also can lead to gene duplication and chromosome evolution, which may have positive consequences for cells. We propose that DNA2, whose activities include degradation of oversynthesized DNA fragments, prevents their insertion into the genome and alteration of the genetic code. The researchers also investigated the origin of the DNA fragments that insert themselves into chromosomal breaks. They discovered that small genes, DNA segments at the end of chromosomes called telomeres, as well as other sequences of inserted DNA had originated from all over the genome. It has been reported that similar insertion of DNA fragments is common in cancer. We think that the mechanism we describe here for DNA2 deficient cells is likely to be observed in many cellular conditions where small DNA fragments are generated and not properly degraded. We think that this is common in cancer but can also occur in patients that are deficient in some components of innate immune system responsible for elimination of foreign DNA. Good day, this is Professor Will, CBMD, PhD. Today, Sunday, December 9, 2018, 19:28 and 6 seconds Eastern Standard Time. A bacterial protein is found to promote cancer. Research suggests that bacterial infections may contribute to far more cancers than previously thought. Date December 4, 2018. Source University of Maryland School of Medicine. Summary. Researchers have discovered that DNA-K, a protein, the bacterium mycoplasma, interferes with the mycoplasma infected cell's ability to respond to and repair DNA damage, a known origin of cancer. Little or no mycoplasma DNA-K DNA sequences were found associated with the tumor, which was fully developed, suggesting a hit-and-run or hide mechanism of transformation, indicating that the damage is done early, but the protein may not be needed once the cancer cells are formed. Currently, approximately 20% of cancers are thought to be caused by infection. Most are known to be due to viruses. Mycoplasmas are a family of bacteria that are associated with cancers, especially in people with HIV. Our work provides an explanation for how a bacteria infection can trigger a series of events that lead to cancer. Of particular importance, the infection did not need to persist and the protein did not need to continuously present in all cancer cells. The study also provides a mechanism for how some bacterial infections can interfere with specific cancer drugs. Researchers utilized immune-compromised mice as a model for analyzing the effect of mycoplasma infection on development of lymphoma. They compared how quickly non-infected immune-compromised mice developed lymphoma compared to mycoplasma-infected immune-compromised mice. The mice were infected with a strain of mycoplasma from an HIV patient. The researchers found that mycoplasma infection caused the mice to develop lymphoma earlier in life than non-infected immune-compromised mice, and that some, but not all, of the cancer cells had bacterial DNA. Finding only a small amount of bacterial DNA in the cancer cells suggested that the infection did not have to persist to trigger cancer. We focus on a protein called DNA-K, which is part of the family of proteins that function as a chaperone for other proteins, protecting them from damage or helping them to fold. However, in this case, DNA-K reduces the activity of important cellular proteins involved in DNA repair and anti-cancer activity such as P53. Thus, cells infected with mycoplasma would not be able to properly repair damaged DNA, thus potentially increase the risk for cancer development. Scientists noted that the bacteria can release DNA-K and the DNA-K enters nearby uninfected cells. The study also demonstrates that by reducing P53, DNA-K can also reduce the efficiency of anti-cancer drugs. Thus, mycoplasma infection could not only trigger events leading to the accumulation of DNA damage and oncogenesis in infected cells, but also trigger cancer-causing events in nearby uninfected cells that took up DNA-K released from infected neighboring cells. We analyzed the amino acid sequence of DNA K from many bacteria and found that the DNA K proteins from bacteria associated with cancer grouped together were different DNA K sequences from bacteria that are not associated with cancer. This raises the possibility that other bacteria have the same cancer-promoting ability. According to a very special researcher, this hit-and-run or hide mechanism mediated by a protein common to many cancer-associated bacteria changes how we need to think about infection in at least some cancers. Furthermore, this provides a basis for understanding how infection can influence the effectiveness of some cancer treatments. Good day, this is Professor Will, CBMD, PhD. Today is Sunday, December 9, 2018, 19:15, 29 seconds Eastern Standard Time. Science News, biologists show inner workings of cellular undertaker. Date, December 4, 2018. Source, Scripps Research Institute. Summary, scientists have deciphered how the proteasome converts energy into mechanical motion that untangles and unfolds proteins for destruction. The findings could help us understand how proteasomes keeps diseases such as Parkinson's and Alzheimer's at bay. The proteasome is like the cellular undertaker for proteins that are no longer needed or pose a threat to the cell's health. As we age, our proteasomes become less efficient, which can lead to a variety of diseases. The proteasome does not readily reveal its secrets and has been a challenging biological target. Although it is one of the largest machines in the cell, the proteasome is nonetheless incredibly small. Imagine looking up at the moon and trying to read a street sign placed on the surface. That's the scale we are dealing with. To further complicate matters, the proteasome structure is very complex, containing a motor and many moving parts. The researchers were able to visualize the proteasome inner workings thanks to the structural methodology called cryo-electron microscopy, cryo-EM. This technology freezes biological complexes in mid-motion, allowing scientists to see many different conformations at the same time. It is like taking a snapshot of a busy freeway at rush hour. The engines powering the cars will all be in different states of firing, but by looking at millions of them, we can figure out precisely how they work. To truly understand how a motor works, you need to observe it while it is running. Other structural methods would require us to shut down the motor or throw a wrench into it to stall it. Cryo-EM allowed us to catch the proteasome red-handed, with pistons pumping as it pulled on a targeted protein for degradation. Specifically, the researchers learned how ATP, the fuel source of the cell, powers movements within the proteasome motor that enable it to pull in proteins through its central channel. Like pulling a tangled ball of thread through the eye of a needle. The proteasome properly positions proteins, threads them, and feeds them as a single unfolded strand to the degradation chamber. Now that the scientists have a better understanding of how the proteasome works, there are significant ramifications for studying neurodegenerative diseases. This has implications for understanding what happens when proteasomes encounter proteins that are very tightly folded, such as the amyloid proteins associated with Alzheimer's disease. This is also important for cancer therapy. Some drugs that are used to treat the blood cancer multiple myeloma are targeted against the proteasome. These drugs target the shredder blades rather than the motor. Now that we understand the motor, we can work to develop a more regulated response and target similar motors found in other cellular machines. Thanks to this research, we've become much better mechanics. Going forward, we'll have a much better shot at figuring out what's happening to our proteasomes as we age, and work out how to keep them running efficiently and effectively like well-oiled machines. Today, this is Professor Will, CBMD, PhD. Today, Sunday, December 9, 2018 is 20:03, 30 seconds Eastern Standard Time. Science News, new strategies may improve CAR T-cell therapy. Date, December 3, 2018. Source, Mayo Clinic. Summary, researchers have developed two new strategies that may improve the performance of chimeric antigen receptor therapy, CAR T-cell therapy, in treating cancer. Reducing toxicity in CAR T-cell therapy. While CAR T-cell therapy has proven successful in treating certain cancers, severe toxicities have limited its widespread application. Researchers say toxicities associated with CAR T-cell therapy include cytokine release syndrome, in which patients experience fever, nausea, headache, rash, rapid heartbeat, low blood pressure, and difficulty breathing, and neurotoxicity. Researchers say some patients undergo CAR T-cell therapy, get sick during treatment, and require a stay in ICU. Researchers also note that deaths related to the side effects of CAR T-cell therapy have been reported. Researchers and colleagues developed a strategy to reduce the severe toxicity associated with CAR T-cell therapy. The strategy involves blocking the GM-CSF protein, which is produced by CAR T-cells and other cells using clinical-grade antibody. When we blocked the GM-CSF protein, we found that we could reduce toxicities in preclinical models. We also were able to demonstrate that CAR T cells worked better after the GM-CSF protein was blocked. Next, researchers used a gene editing technology called CRISPR to generate CAR T cells that did not secrete the GM-CSF protein. Researchers say these modified CAR cells were more effective than regular CAR T cells. Based on their findings, the research team is proceeding with the Phase 2 clinical trial of the GM-CSF blocking antibody during CAR T cell therapy. If the trial results are consistent with earlier findings, the therapy could become a standard of care during CAR T cell therapy at Mayo Clinic. This research also is published in Blood, improving response rates for CAR T cell therapy in B-cell lymphoma. In CAR T cell therapy, physicians remove and modify a patient's T cell to recognize and fight cancer. Once modified T cells are reinfused into the patient, where they seek out and ultimately kill cancer cells. Researchers say response rates for CAR T cell therapy vary by disease. For example, in B-cell acute lymphoblastic leukemia, response rates of more than 90% have been seen, compared to response rates of 10 to 30% for treatment with conventional chemotherapy. In other blood cancers such as lymphoma and chronic lymphocytic leukemia, the response rates for treatment with CAR T cell therapy remain low. To improve the effectiveness of CAR T cell therapy in these cancers, researchers and colleagues developed a strategy to combine CAR T cell therapy with a drug that targets a protein called AXL. The protein is present on the cancer and within the cancer's environment. The drug called TP-0903 not only kills cancer cells, but also it enhances the potency of CAR T cells in attacking cancer cells and potentially lowers the toxicity associated with CAR T cells treatment. While more research and clinical trials are needed, researchers say we believe the later effect may eventually be utilized as an innovative approach to augment the efficiency of CAR T cell therapy and extend its use to other B-cell cancers. Good day, this is Professor Will, CBMD, PhD. Today, Sunday, December 9, 2018 is 2021, 30 seconds Eastern Standard Time. Science News. Study discovers 40 new genetic variants associated with colorectal cancer risk. Collaboration involved researchers from more than 130 institutions globally. Date December 3, 2018. Source: Fred Hutchinson Cancer Research Center. Summary: the most comprehensive genome-wide associated study, or GWAS, of colorectal cancer risk to date has discovered 40 new genetic variants and validated 55 previously identified variants that signal an increased risk of colon cancer. Researchers have identified the first rare protective variant for sporadic colorectal cancer. Sporadic colorectal cancers have no known familiar syndrome and account for the vast majority of colorectal cancer cases. Together, these findings are a significant step toward creating personalized screening strategies and better informing drug development for colorectal cancer. Understanding the genetic architecture of colorectal cancer will revolutionize how we assess risk and treatment for this disease, which is the second most deadly cancer in the U.S. These findings also illuminate what the study authors call a missed opportunity in drug development. The study identified several loci, the physical location of the gene on a chromosome near proposed drug targets, and genes and pathways not previously known to be causally linked to colorectal cancer. Using GWAS results to inform cancer drug development, the authors believe could improve the drug development success rate and even lead to chemoprevention drugs for high-risk individuals. There's great potential in using GWAS results to inform target discovery for anti-cancer drugs. For diseases like type 2 diabetes and heart disease, the GWAS approach drives the discovery of new biology and potential drug targets. Search for new targets for cancer therapy has been limited to focus primarily on the molecular characteristics of cancer cells. We think there is a huge opportunity using the GWAS approach to inform drug development for colorectal cancer. Researchers from more than 130 different institutions contributed to this study, many by sharing data and biospecimens that allowed the team to analyze the genomes of more than 125,000 individuals, of which there were 58,131 colorectal cancer cases and 38,296 control participants who had not developed the disease. The number of participants in this study is nearly double that of previous analysis of this type. Large-scale whole genome sequencing studies have discovered millions of genetic variants that have yet to be examined systematically for association with disease. The population reference panel has sequenced data from more than 32,000 individuals. By coupling this strategy with a custom-designed genotyping chip, we were able to robustly identify a rare variant association signal and multiple additional signals involving lower frequency variants. Previous GWAS for colorectal cancer risk only looked at common genetic variants. In contrast, This study involved whole genome sequencing of more than 2,000 individuals and was designed to examine the contribution of rare genetic variants to colorectal cancer risk. With this study, we brought the known number of risk variants for colorectal cancer to nearly 100. Next, we are broadening the study population to include people from diverse ethnic backgrounds. This will give us a more complete understanding of risk across the entire population. The authors acknowledge a bias in the study sample, of which 91% were people of European descent. It is critically important that we increase diversity in our future studies, because premature use of these findings to inform screening guidelines could exacerbate existing racial disparities in colorectal cancer screening and survival rates. Genetic variants occur from differences in our DNA. Most variants are believed to be benign. Some are known to be associated with various diseases, and the significance of many others are unknown. While individual genetic variants have little impact on disease risk, several combined variants can become clinically relevant, and this could have an impact on future personalized screening recommendations. To evaluate a person's full risk profile, genetic risk factors need to be combined with other epidemiological factors such as diet, weight, and exercise. In some cases, a low genetic risk score could be skewed by unhealthy lifestyle factors to yield a high overall risk profile. This type of work is currently conducted by the team, which not only collected genetic data from all studies, but also harmonized many clinical and lifestyle risk factors across more than 70 studies. Furthermore, the research team believes there may be hundreds of other genetic variants contributing to colorectal cancer risks that have yet to be identified.